Welcome everyone and Happy New Year. My name is Michael Lemke. I am the Senior Director of Science and Industry Affairs at the Alliance for Regenerative Medicine. Welcome to the first Aging webinar of 2022, part of a continuing series. Today's webinar will be on comparability, a topic that I'm sure is of great interest to all of you. Before we get started, just a little bit of background for those of you who might not be familiar with aging. Aging is a best practices guide. It was first conceived of at an ARM-CMC workshop in 2017. It was recognized by our members at that time that issues related to CMC had the potential to result in major program delays. And I think, unfortunately, we have seen some of that come to be true. And this is in part due to an absence of best practices in the industry. So inspired by another effort in the monoclonal antibody world called AMAB, ARM decided to pursue producing a best practices guide or case study of the manufacture of AAV vector using a triple transfection process in an HEK 293 cell line, importantly employing quality by design principles. We are in the midst of a webinar series, an educational series on the aging document for our members. You can find out about these webinars on our website at the link listed here. I believe we have 2 more still to go, which we hope to finish out in the first quarter. And then we will have a half-day workshop focusing on the entire aging guide a little bit later in the year, date TBD at this point. For anyone who wants to read the full document or access it, you can view it and download it on ARM's website at the link shown here on this slide. And these slides will be made available to you after the webinar today on our member portal, along with the recording of the webinar. With that, I'd like to introduce today's speaker, Mo Hadaran, an expert in comparability. Dr. Hadaran joined Parexel International as the Vice President of Technical in December of 2018. Prior to that, he worked as Acting Branch Chief in the Division of Cellular and Gene Therapy in the Office of Tissues and Advanced Therapies, and as a CBER-wide subject matter expert providing advice to ADRM in the Office of CBER, Director on special subject matters related to CMC policies and regulatory review management processes. He has 8 years prior experience as a biologist and a master reviewer in OTAT, and a facility reviewer and inspector in the Division of Manufacturing and Product Quality. Mo is a multidisciplinary academic and he has an industrial background in basic and applied cell biology and innovative cell therapy and tissue engineering product development. He has hands-on industrial experience in the manufacturing of cell therapy and tissue engineered products for 15 years in small and large biotech companies. Dr. Hadaran holds a PhD in biochemistry from the University of South Carolina and received his formal training at the National Cancer Institute. He holds 25 issued patents, 54 pending patents, and his work has appeared in more than 50 scientific publications. Mo, welcome, and with that, I will turn the microphone over to you.
I'm very pleased to be here. Thank you for your invitation. This slide is a combination of probably 10 years of thinking about this subject. I started thinking about this perhaps 10 years ago when my old boss at FDA asked me to go give a presentation on what are the essential elements of a good comparability study. So it's been 10 years in making. Today, I'm going to try a little bit about, talk about a number of things that have experience in this field. Obviously, there has been a lot of questions. Comparability is a major issue and it's complex. It's case by case and product dependent and so forth. But today, I'm going to a little bit try to kind of give the high level sort of perspective of comparability. I do not work at FDA, obviously, anymore. So my views are my personal perspective. So as a background, there is, as shown, the first slide is really, I'm going to talk about why establishing comparability after introducing major changes is very important in cell and gene therapy field. Talk about what type of changes require comparability. And it seems like somebody's advancing my slides, but it's not something I'm doing for sure. And then what are the risk factors affecting product comparability, challenges in that area, and how to deal with manufacturing changes, and talk about essential elements of comparability studies at the end. So we all know, the question, and this question, and the answer to this question: why manufacturing changes are so critical in cell and gene therapy. Obviously, as you know, products are not specified, unlike biotech products that are highly characterized. Most of the product in this field are not. And essentially, also the science tells us that minor changes in incubation, cytokine concentration, the environment, etc., has a significant effect on property of many of these cellular products. Is there a way to minimize advancing by any chance? Because my slides keeps advancing automatically. But at any rate, and also the product knowledge is limited, it's incomplete. Most of these products have incomplete understand knowledge. Most of the product manufacturers have incomplete knowledge of the critical quality attribute and critical process parameters. This slide that deals with some number of questions that I'm being asked often by manufacturers and IND holders. The first question I wanted to answer is, what type of changes require comparability studies support? The answer is outlined here, is essentially summarized. And generally, anytime sponsors want to rely upon clinical data generated product manufacturers before major manufacturing changes are introduced, and combine that data with the clinical data after that change is introduced. And in this combination typically triggers the need for comparability studies. So essentially, if you have some pre-existing clinical data with an old version of the product and you want to make the change and combine the clinical data before and after, this requires assessment of the comparability for the product, particularly if this is done late phase. The other question I've been asked is, can comparability be established for change in manufacturing based on just meeting a specification? Generally, the answer to this question is no. And of course, I've been asked, what's the best way to engage FDA in this area of establishing comparability? For minor changes or changes early during development, you may not need to have early engagement of FDA, but if you are introducing major changes prior to pivotal studies, during or after, I highly recommend engaging FDA in that process. So obviously, we don't have a clear guidance for establishing comparability for products that are being developed during the early life cycle. But I wanted to point out that there's a very good guidance document for post-approval changes that I wanted to point out here, and that is chemistry manufacturing control changes of the approved application, certain biological product, which is a draft guidance, and I believe it has been finalized. This guidance document covers cell and gene therapy product and provides some examples of what manufacturing changes trigger comparability studies. Essentially it defines changes as minor, which are annual reportable, moderate, which requires CBE 0 or CBE 30, or major changes that require prior approval supplement. This slide sort of highlights some example. I'm not going to dwell on a number of these examples that falls in each category, but suffices to say, if you have a major manufacturing changes, and this is done post-approval, that's being discussed in this guidance document and requires prior approval supplement, which are 4-month supplements. And so this means that the changes cannot be implemented until these changes are approved. Oh my God. My slides, can I reshare my slide because it keeps advancing?
Yeah, if you'd like, we can advance them for you.
Mo, just jumping in real quick, you have a timer on your slide, so if you minimize your screen, I can tell you how to, or I can share the slides, whichever you prefer.
There's a timer on the, where is the timer?
Yeah, so if you click the slideshow tab at the top, toward the end, fourth from the end, if you go to the right.
No.
Sorry, yes,
Yeah,
insert, design, transit, animate, and then go to slideshow. So 3 more over. Animate, go, 2 more over. Slideshow.
Okay. And do.
And then if you click setup.
Right.
And then uncheck that box that says use timings.
Oh, geez. Oh, thank you.
That should fix it.
Okay, that's great. Thank you.
You're welcome.
I think I appreciate your help because I wasn't aware that I was timing myself. So so the next slide is really highlights what are the risk factors affecting product comparability, and I hope I'm still sharing my document. And of course, you know, we know the knowledge of CQA and CPPs are very critical in establishing product comparability, but also it is important to note that essentially the requirement for comparability study is really phase dependent. By that I mean that the robustness of comparability studies for changes during early phases is different than what is required to be established for late phases. And this is essentially caused by the situation that we are in based on the limited knowledge of the CQA and CPP. The risk that minor changes or major changes affect product quality is somewhat unknown, and thereby the agencies like FDA have set the bar higher when manufacturing changes are introduced late during the product development cycle. So essentially the bar is phase dependent. If manufacturing changes done before pivotal studies relative to manufacturing changes after pivotal studies, obviously the risk would be higher if manufacturing changes are introduced after pivotal studies. So this is a reason why I highly encourage manufacturers to introduce major changes early during development as much as possible, because the requirement and the bar that needs to be achieved is lower than if you may introduce those changes late during the development, i.e. During the phase 2 studies or post phase 2 studies, or in the middle of the pivotal studies versus introducing manufacturing changes after pivotal studies. And so for this reason, it is very important to denote that the robustness of analytical comparability and the requirement increases as you go toward late phases during the development. So process changes are obviously inevitable in this arena, and essentially sponsors are responsible to report those changes. During IND phase, major changes have to be reported as amendments. Sometimes these major changes are accepted by FDA based on limited comparability studies, but in some other cases, FDA may ask for additional information. It's always risk and science-based. Some of the major changes are outlined here that I see in this field. For example, the site of manufacturing may change. There's a change of major critical reagents, automation, and scale out to scale up. Product comparability is intended, again, to demonstrate that the changes in process does not adversely impact product quality, as you know. And it's really, the bar is not to show that the product before and after change are identical, but there is expectation that you establish that they are similar. Although ICH-Q5E is not necessarily cover cell and gene therapy products, some of the guidance provided in Q5E is relevant to ATMPs, but there are some other documentations that have been published. For example, question and answer comparability consideration for advanced therapy medicinal product that was published by EMA that could be consulted. But for any major changes during late phase, I always encourage manufacturers to reach out to regulatory authorities and to engage in discussion of what the comparability studies look like, and to be able to proactively deal with some of the questions they have in comparability study protocol. One of the things is challenging is in case of comparability study for cell and gene therapy is really, again, is our limited knowledge of CQA and CPP, critical quality attribute and critical process parameter. I know Agene had another chapter that talks about this, which is highly relevant here. And again, additional factors is in this area is small sample size of the product manufacturers before and after change. Sometimes it is impossible to actually do a side-by-side comparison. And some of the batches of manufacturers are very difficult to produce and expensive. Therefore, there are some technical and business consideration in this area that has to be factored in. For gene modified cells, obviously the starting material and patient donor material variabilities and other consideration. And historical data, sometimes not available actually, or there's a limited amount of historical data available, so that creates some challenges. And the statistical methods also sometimes inappropriately used for determination of the product comparability. And for those reasons, establishing product comparability is very challenging and very time-consuming. How do we establish product comparability in this space? Of course, if the product knowledge is rich, by that I mean you have a rich knowledge of the product critical quality attribute and CPP. Always to talk about the need to do essentially a side-by-side comparison of those critical quality attributes for the product using appropriate statistical method, etc. But this is not the reality in our situation. And in most cases, there's incomplete knowledge of the CQA and CPP. Again, I bring it up. And in those cases, the matrix-based approach is encouraged by FDA. The matrix-based approach really looks at some of the critical quality attributes, candidate critical quality attributes side by side. It looks at the determination and measurement of those attributes in process or in the final drug substance or drug product. It also talks about manufacturing yield, looking at the control of the key process parameter, risk assessment, and sometimes QBD approach can be applied. But essentially, it's a matrix-based approach in which you can really compare all relevant product attributes and before, after change, looking at qualified assays that are necessary for not only release of the product, but also characterization of products. So extended characterization is a very important part of establishing product comparability for cell and gene therapy products. Just quickly, I want to just put a plug for really the importance of identifying CQA, CPP, and KPP for any product manufacturers. Obviously, these parameters are very important. They are important because essentially the higher knowledge you have of these parameters, the easier it is to establish product comparability and convince the regulatory authorities in this regard. Essentially, these attributes are outlined here. I don't want to talk too much about them, but it is very important that early on during the development that candidate CQAs are identified, and really one looks at how these attributes can determine or affect product safety and efficacy. And in order to do that, always preach the following slide, and that is, it is very important that that each investigator or company has a focused program which is systematic and iterative approach of identifying product specific CQAs. And I do realize that this is a difficult task to accomplish for a cell and gene therapy product, but it is an aspirational goal that FDA nowadays talks about. And the process essentially starts with candidate CQAs, development of assays that are qualified to measure those CQAs, and defining specification for those CQAs in the context of experimentation, and correlating those attributes with clinical outcome. And this obviously is an iterative process, and you will have very limited knowledge early on during the phase 1 clinical study as compared to phase 2 or phase 3. But there is a expectation that as you go throughout the development that you acquire more knowledge of the CQA and CPP and how relevant those quality attributes are to product quality and product efficacy. And by doing this, essentially that expectation can be met. It is very important that nowadays that FDA has the expectation that you identify candidate CQAs and CPPs for any product and have an iterative process in order to be able to correlate those product attributes to product bioactivity such as potency assay and eventually the clinical outcome of each product. Understanding that it is very complex, but the process has to be in place in view of product manufacturing and in view of the challenges that one will see during product development. So I'm going to give you a couple examples of changes for gene therapy product. I included one example of a gene-modified cells. In this case, you can consider this as a CAR-T product. Major process change has occurred in this case. So the comparability approach is really defined on the slide, the section to the right. You would be required to do essentially a side-by-side comparison using same donor materials. So in most cases, healthy donors are accepted, and a split strategy is preferred to minimize donor-to-donor variability. N of 3 would may be acceptable, and obviously, if you do implement some of these changes late during the phase, essentially, it is important to have a discussion with the agency about your comparability study protocol prior to implementation of this study. So this gave an example of a change in, let's say, process change or site change for a product that is gene-modified cells, and some of the practices that are accepted today in order to establish product comparability. The second one talks about manufacturing of a V product. Obviously, in this space, there's a huge push in really scaling up manufacturings from the academic side to more commercial side, and this transition requires establishing comparability study. And in essence, this can be established in most cases by a side-by-side comparison. Sometimes side-by-side comparison may not be feasible, and of course, in those cases, product manufactured after change have to be compared using well-defined assays to retains or lots manufactured prior to the change. And this is why it is very important in AAV manufacturing that the retains are used, or reference materials is established based on the material that are used prior to the change, because that material can be used as a reference material for comparative purposes to the material that is manufactured after the change. In most cases, N of 3 is acceptable. And of course, the process when you get to late phases that are used for comparability studies has to represent the commercial scale manufacturing. This slide essentially summarizes some of the comparability study point to consider. For comparability study protocol, you have to have a description of proposed changes. You have to have a risk assessment accompanied with that, have a rationale for proposed changes, and there has to be a well-defined comparability study design. There needs to be detailed information of proposed changes and potential impact on product quality. There has to be a ranking of critical quality attributes for product safety, purity, and potency, which is really deals with what needs to be measured comparatively. Comparative assessment has to occur before and after change. Side-by-side comparisons are always preferred, and using the same donor material if possible for gene-modified cells. And there has to be justification of well-defined acceptance criteria, predetermined acceptance criteria for establishing analytical comparability. And the document has to have detailed analytical procedure, sampling plan, and statistical method. And in most cases, after the change is implemented, if it is acceptable, the change has to be introduced, implemented, and the new process in some cases have to be validated for commercial manufacturing. The stability has to be established with product manufactured after the change as well. And the protocol also must include a conclusion section to determine if the products are determined to be comparable, and future commitment, if any. in the protocol has to be included. Now, I'm going to talk about the description of the changes, and talk about some of the changes that you may envision, whether it's a major, minor. Categorization of these changes very important in your comparability study protocol. And in most cases, in situation where manufacturing change are incorporated, there are a number of process changes are included, plus the site of manufacturing is changed. These are accumulatively could be considered as major changes, but sometimes, for example, some of the changes by themselves could constitute a major change as well. These are included here as sort of a guide for assessment of the of whether a change is minor, moderate, or major. But it is very important that you have made that assessment in your comparability study protocol in order to appropriately study the comparability study for the product manufactured before and after the change. I don't have a lot of time to talk about some of these changes, but I have seen some changes that go beyond comparability. Sometimes it could trigger the requirement for a new IND that I have outlined some of those here, and you can have a discussion with the agency about those changes as you go forward. This slide kind of talk about how do you establish comparability. We talked about comparing side by side critical quality attributes. It really deals with what has to be measured. Sometimes I see comparability being established by just testing and assessing quality of the product based on release specification, and that alone is used for establishing comparability. But comparability is not an exercise of really trying to meet the release specification for a given product before and after change. It is about comparing the critical attributes before and after change. And so this talks about what is critical to the product's safety and efficacy. The knowledge that criticality is important in your submission, and as part of the discussion you would have. So for AV products, for example, you can see that the ratio of full to empty has become very critical for product quality. The potency of the product, whether it's measured by RNA expression or protein, are critical. So these are critical attributes that have to be measured. The impurities that you had with the host cell, DNA or RNA, could be critical quality attribute. And so it is very important to focus on critical attributes when comparability studies are being designed. And there should be more emphasis on more critical attributes than emphasis on less critical attributes for product quality. In all cases, from the safety perspective, there are some mandated tests that have to be implemented, such as endotoxin, mycoplasma, and sterility. Those are mandated tests that you need to perform while you're implementing comparability studies. But the essential of a comparability study has to rely on the measurement of the critical attributes that can be linked with product efficacy. And some of these attributes are outlined here, such as potency and vector quality, etc., or the percent full to empty, or the strength of the product based on the ddPCR. The other aspect is the use of statistics, and of course that is probably the most challenging because of the sample number being very small. In this situation, obviously comparability study can use a variety of techniques that are outlined here. Some of these techniques are described very well in aging chapter on comparability. I refer you to the statistical strategy for comparability assessment that was written by Ali Chang, and it has a very comprehensive discussion of the statistical methods that can be used in this area. But without going through each one, which relies on minimum max type of strategy or tolerance interval or other means, I just wanted to note that recently FDA has been prescriptively requesting TOAST as a test of equivalence for comparability studies. So these two one-sided t-test is preferred from perspective of comparability studies from FDA perspective based on what I have seen recently. Of course, there are other methodology that can be used, such as simultaneous prediction interval that can be reviewed in the aging chapter on comparability as well, that I refer you to that. But essentially in this area, I definitely highly encourage the use of TOAST, two one-sided t-test, and I would try to avoid inappropriate use of t-test for establishing comparability. The other aspect is for comparability studies, you have to have a predetermined acceptance criteria, particularly if you want to introduce a change late phase or mid pivotal studies or after pivotal studies. Predetermined acceptance criteria, as mentioned, can be configured using SPI or other methodology, but really looks at the historical. You need to have a rich amount of historical data, and one has to consider inherent variability in the assays that are being used. So there are several methods that are being implemented, but in all cases, you have to take a look at historical data, assay variability, look at the statistical method, and come up with the predetermined acceptance criteria prior to implementing the comparability study protocol. And there has to be sufficient justification for these predetermined acceptance criteria. The other aspect is the need for process validation if the criteria for comparability is established. In most cases, the product that is manufactured based on a new protocol or new process or at the new site, it still needs to be, that process still needs to be validated or revalidated after a change is implemented. This slide talks about some of that requirement, and the requirements are essentially really geared at establishing that following changes in manufacturing, that you can make that product consistently. And if the consistency of that product is not established, then there will be essentially a bottleneck for BLA approval. So although comparability study can be used to introduce major changes, it is very important that it is also accompanied with process validation if it is intended to be used for commercial purposes. And this slide talks about a little bit about process validation and what is expected at the level of commercial scales. I'm not going to belabor the point here because we have some time limitation, but I wanted to focus more on the stability post-change. The agency has an expectation of establishing product stability post-change. Real temperature, real-time type of stability is needed. Some limited information should be provided as part of comparability study. And in some cases, accelerated stress stability is may be needed, but in most cases, it is not required. Of course, the use of accelerated approach required understanding of relationship between accelerated and real temperature storage, and helps define whether an attribute is or the assay that is used to measure an attribute is a stability indicating, and it really gives you a deep sense of knowledge in regard to product quality. So although it is not required, accelerated stress stability for product like AAV is very helpful to have post-change. For products such as gene-modified cells, accelerated stress stability is not practical and it's not meaningful. A couple of slides about the use of well-defined assays that are qualified versus validated. For comparability study, it is highly recommended that all the methods that are used to be qualified at least or validated if a change is introduced late phase during the development, let's say after pivotal studies. And this slide highlights the differences between qualification and validation in a sense. And it is important to point out that as part of qualification, it is expected that the assay is sensitive, accurate, reproducible, and this is a requirement for early phase, and validation has additional component of robustness and ruggedness that is important to keep in view. And these are required for obviously licensure as well. But for purpose of comparability, if a product before and after changes compared, or the product after the changes compared to the reference material, it is very important that the assay that are used are at least qualified and most ideally validated. And this is an essential part of the comparability studies for AAV products, in my opinion. And the availability of reference material, products that are retained under stable conditions prior to that change is very important as element of establishing comparability study. This probably one of my final slides. So the major consideration for comparability is to define what is the change, what is the level of risk that change impact product quality, and why the change is introduced. These have to be discussed in your comparability protocol. And of course, it is very important that you also discuss in the context of product development cycle. The expectation of establishing comparability is very phase dependent. And it is important to know that the more reflective CQAs are of clinical outcome, product potency, the easier it is to establish product comparability. One additional note, the more robust your comparability and based on analytical parameter is, the lesser likelihood that FDA requests for clinical data and bridging information. So there is a reciprocal relationship between the robustness of your analytical comparability and the requirement for additional non-clinical and clinical studies for bridging. And that has to be viewed in the context of your strategy to introduce manufacturing changes. The essential aspect of comparability studies, again, are described here. In a protocol, you would describe the proposed change. You include the risk assessment, rationale for those changes. You will have the well-defined comparability study design. You will talk about critical quality attributes and rank them accordingly, focusing on the highly critical attributes that have to be measured comparatively. There will be a side-by-side comparison, ideally, of those attributes using a well-defined predetermined acceptance criteria, and also statistical method and sampling plan. There's a requirement for predetermined acceptance criteria when these changes are introduced late phase. Post-change for commercial purposes, process validation may be required, and definitely stability of the product manufactured using the different process or at a different manufacturing facility would be needed to be accompanied. So summary and conclusion. As you know, manufacturing changes are inevitable, obviously part of the process. We see that transition from phase 1 material to commercial material. Often there is this involves change of facility, change of the process, change of producer cell lines. These are all major manufacturing changes that have to be accompanied with a comparability, well-defined comparability studies that looks at analytical similarity. If possible, avoid introducing major manufacturing changes late during product development cycle. I hope I convince you that it is important to introduce these changes early on during the product development cycle rather than late. The later these changes are introduced, the higher the risk of not being able to establish comparability based on analytical results only. If manufacturing changes are introduced late, you should have well-defined comparability studies. And I highly encourage manufacturers to think about comparability study very early during the development. Develop comparability protocol based on knowledge of product and based on your knowledge of CQ8, etc. And establish a template for comparability studies early on during the development to be proactive and be ready for conducting some of these studies. In most cases, it is important to note that product comparability cannot be established based on meeting the release specification. Comparability protocol is a prospective plan, and it has to be accompanied with what discussed in this series and in this meeting, looking at the essential of comparability studies. I outlined about 8 or 9 essential components of comparability that has to be followed. And for late phase, I highly encourage you to discuss your planning with the agency prior to implementation, get their concurrence overall regarding the comparability product, comparability protocol, and have a collaborative and discussion with the agency about the adequacy of your approach to comparability studies prior to implementation of those studies. Thank you so much, and that ends my talk today.
Thank you, Mo, for that very thorough presentation. We have some questions from the audience already, and I would invite the attendees to submit additional questions using the Q&A window. Mo, I'm going to start off with a question of my own. Why in the cell and gene therapy field is comparability, and in particular pre-approval comparability on products that aren't approved yet, such a big issue?
Well, you know, for scientists who've been in this field for a long time, like me, you know, having a background in doing work in laboratory setting, and you know, to be honest with you, the challenge is that there is a precedent that very minor changes in manufacturing can cause a profound change in product quality. So there is a precedent for that. That really essentially increases the risk. That's one. And the FDA, as you know, has a very science-based approach. Half of the regulators at FDA do work in laboratory. And they understand that if there's a minor change of a cytokine, it changes the behavior of cells profoundly and dramatically. And so they are exposed to example whereby a minor change perceived as minor could have profound effect on biology of a cell. The second part is that, again, the CQA and CBP historically have been ignored in this field. And now FDA emphasizes the importance of establishing at least some candidate CQA and CPP in order to define what is critical to product quality, i.e. the potency of the product's bioactivity, and hopefully relating that to clinical efficacy, as I discussed. So those are the two components that are unknown, and that therefore there's elevates the need for establishing analytical comparability in this field based on a matrix-based approach.
Thanks, Mo. Great answer. And just for the benefit of the attendees, development of CQAs and mapping CPPs to CQAs is a topic that's covered in detail in the AGene guide. I have some questions from the audience. Are there opportunities to apply ICHQ9 and first principles to reduce comparability analytical testing to only the CQAs that may be affected by a process change, rather than a full comparability panel, which might include release, characterization, force degradation, etc.?
Yeah, I tried to point that out that in the section that about what needs to be measured. This question relates to that. What is critical to be measured that really looks at product quality in view of product bioactivity and clinical efficacy? If you have a good package of knowledge around that, so therefore you can focus on those attributes for purpose of comparability studies, because those are the most critical to product quality. And unfortunately, in most cases, that is not available. And therefore, the industry is kind of, the default mechanism is to test what is on your release specification. But it is important to point out that characterization results become even more important in cell and gene therapy, field, that sometimes the release specification. So one has to have the perspective of identifying critical quality attributes. I think that's given. And if you have a robust package there, then you can limit it to those quality attributes in your study design. That is a possibility. Yes.
Thank you. Next question. Can you speak to circumstances in which comparability using less than 3 GMP batches or a combination of GMP and engineering runs may be acceptable?
Right. So I think the agency has shown some flexibility in that area of what do you call batches, whether the engineering batches can be used or not, and so forth. It is possible to have justification for the sample number, even maybe less than 3, maybe 2. Although it is very important to have that N of 3 for statistical purposes, but it is not impossible to have less than that. Let's say 2 GMP run and 1 engineering run or additional development run. But all of those have to be done using the new process, obviously, and have to be representative of that new process if the process is changed. Or they have to be done at a different facility. If the facility is changed, obviously, the process has to be representative of the manufacturing, commercial scale manufacturing. The other thing I want to point out that's distinct in cell and gene therapy is we don't have scaled down models like in biotech environment. And that's why it is always, virtually always, comparability at the large scale has to be done with actual batches. The development run, engineering run at commercial scale, because lack of a scaled down model that exists in this area versus what appears to be available in biotech environment. So those are the challenges. So yeah, development, engineering run can be included, but have to be representative of that process.
Got it. So here's, I think, a good question. At what stage of manufacturing should you be studying the product in the comparability study? After each unit operation, at the drug substance stage, at the drug product stage, all of the above?
Yeah, so I recommend doing some in-process monitoring using the same attributes that are critical, as well as the drug substance and drug product. Now, that would be the more comprehensive approach, but sometimes realistically, this is not practical. And it is important to kind of prioritize if you introducing changes in the process. Obviously, for a V product, the SDP, comparative measurement of attributes between DS and DP is more important than in-process testing. But in essence, in-process testing is part of a comparability study, should be part of it, but in some cases it may not be practical. But I do recommend testing some of those attributes in process as well in order to establish comparability, if feasible.
So unfortunately, we had quite a few questions that just came in. We're not going to be able to get to all of them, but we will save those questions and try to get back to attendees later on with the answers. We'll post those on our member portal, but maybe I could just answer one more question here, or I could ask one more question of you. When analytical comparability won't suffice, can you comment on what might be a reasonable approach to using non-clinical testing? Or would you expect that normally in this situation, clinical comparability data will be required?
So I made a statement, general statement that the better your package is, the better understanding of the comparability is using well-defined, qualified, validated assays and CQAs that are relevant to product efficacy. The less likelihood that they would ask you for clinical bridging data. It also another factor is when you introduce those changes, like I definitely think if you introduce changes post-pivotal study, that is definitely an invitation to ask for additional clinical bridging data. So you have to consider when you want to introduce a change. I've seen situation where a company before pivotal studies have decided to change their manufacturing, but for pivotal study, they use the process that is not representative of commercial manufacturing. You increase your risk by, introducing commercial scale manufacturing after pivotal studies that uses less than commercial scale manufacturing. So when you introduce change, is going to impact this relationship, and your product knowledge also impact this relationship as well. But there's no set formula for this. It's product specific, and it has to be, it is case by case, to be honest with you.
Okay, so obviously the timing of the change opposite when your clinical studies were conducted is critical. So,
yeah, I think the main thing is when you want to, I discourage introducing changes after pivotal study is completed. And this increases the risk of analytical comparability not be sufficient. As a reviewer, I really avoid making assessment in that phase, because often the clinical reviewer asks CMC reviewers, do you think these products are comparable based on the analytical similarity? And that's a difficult question to answer. And therefore, one has to be aware that when it is very difficult for a CMC reviewer to make that assessment, the likelihood of needing additional clinical data is raised as a result.
Good, that's great advice. So we're over time, actually, so apologies that we didn't get to all the questions, but we will pull the remaining questions from the recording, and I'll circle back to you on those later, Mo, and we'll make the answers available on our member portal, along with the recording and the slides from the webinar. Mo, I just wanted to thank you once again for participating and for your contribution to aging, and I want to wish everyone a good day and stay healthy and Happy New Year.
Thank you so much. Thanks for your invitation, Mike. Take care. Bye-bye.